It might take a little bit of force to break this up. Usually, the fresher a body is, the easier it is for me to work on. Mortician Holly Williams says by lifting John's arm and gently bending it at the fingers, elbow and wrist, well, we can monitor and modify the chronology of death in the chemistry lab using our knowledge of kinetics, specifically determining the exact time of death and protecting evidence after death. When determining the time of death, investigators typically use two methods, the rate method and the concurrence method.
The rate method.
By comparing the presence or absence of an indicator in the corpse with the known behavior of such signs, this method determines the time of death.
The concurrence method.
By analyzing events that take place at or close to the time of death or provide information that suggests a time period for the death event, this approach estimates the time of death. You'll see that the word estimated is used to define the technique in each sort of determination. This is due to the fact that, despite all scientific developments, determining the time of death remains one of the least accurate methods we have. For it to be an exact science, there are just too many variables that can skew the results. Collecting the body, gathering any nearby evidence, and gathering data about the deceased are the initial steps in determining the time of death. Let's go through each technique and talk about how it's used to establish the time of death. Before proceeding further, if you are new to this channel, then please subscribe to this and hit the like button. I am sure you will get some interesting videos in this channel. Now comes to the video.
FLORA and FAUNA.
Insects and plants are frequently used to estimate the time of death. The length of time a body has been present at a crime scene can frequently be determined through observation. As an illustration, grass that is covered by a body progressively wilts and dies. A botanist can tell you the time of death, if the individual was slain there, or the time the body was placed if they were killed somewhere else, based on the rates at which particular varieties of grass die when covered. Insects in particular quickly infest an exposed body, and maggot formation has a well-established chronology that can be used to estimate the time of death. If the flies get access to the body right away after death, taking samples of the maggots present to an entomologist will give you a reasonably accurate time of death.
Stomach contents.
Based on the degree of digestion, the food recovered in a victim's stomach can provide an approximation of the time of death. If a victim's stomach is mostly full with undigested food, the death most likely happened an hour or two after the meal. The death probably happened more than 4 hours after eating, if the stomach is empty.
Drying blood.
Blood is a complicated mixture of several cell types, nutrients, and cellular waste, although water makes up the majority of it. If we are aware of the environmental factors, such as temperature, humidity, airflow, surface area, etc., blood will dry at a predictable rate as it is an aqueous fluid.
Rigor mortis.
All of the body's muscles instantly relax following death. Due to an accumulation of acid in the muscle tissues, the body gradually stiffens during the following 24 to 48 hours. This stiffening process, known as rigor mortis, occurs at a fairly known time, making it possible to predict when someone will pass away. In general, if the body feels warm and no rigor is present, death occurred under 3 hours before. If the body feels warm and stiff, death occurred 3 minutes 8 hours earlier. If the body feels cold and stiff, death occurred 8 minutes 36 hours earlier. If the body is cold and not stiff, death occurred more than 36 hours earlier. Due to the wide temporal ranges it covers, the use of rigor mortis as a time of death indication is less than optimal. The windows may differ by up to 24 hours. In addition, a number of factors can have a significant impact on the timing and commencement of rigor, including temperature, illness, activity before death, physical conditions where the body are found.
Livor mortis.
Blood settles in the body as a result of gravity, which is known as lividity. Livor mortis begins to manifest 2-4 hours after death, is non-fixed or blanchable up to 8-12 hours after passing away, and is fixed or non-blanchable after that. When you push your finger on your skin and see a brief white patch, this is blanching. The pressure of your finger forcing the blood out from that spot for a few seconds is what causes the skin to become lighter, until the blood rushes back once the pressure is released. The area lacks color due to a lack of blood. To further ascertain the time of death, investigators would press their finger into a pool of blood and check to see if the region is fixed or not. Usually, this procedure is carried out at the crime scene.
Algor mortis.
The cooling of the body after death is known as algor mortis. Blood circulation helps to keep the body at a normal temperature. Circulation stops when the heart stops, and the body starts to chill. The average body temperature, which varies but is widely believed to be 98.6 degrees Fahrenheit. Body temperature is perhaps the method mentioned the most frequently on television and in literature to predict the moment of death. However, it is by far the least dependable because of the variety of outside influences that might affect it, such as body dimensions, posture, clothing, ambient temperature, air movement. The general equation used to measure time of death based on body temperature is the Glaser equation, which is 98.4 minus measured rectal temperature divided by 1.5 is equal to approximate hours since death. This means that the body drops approximately 1.5 degrees for each hour after death.
Decomposition.
The well-known stages of decomposition can be utilized to estimate the time of death, particularly in cases when the body has been missing for weeks or even months. The body decomposes mostly through putrefaction and autolysis. Digestive enzymes in human cells break down proteins and carbohydrates through a process called autolysis. The main factor causing tissue deterioration is putrefaction, which is brought on by bacterial activity. 4 to 10 days following a death, putrefaction begins. Putrefaction is primarily responsible for a dead body's changing look over time, which are bloating, green discoloration of abdomen, marbling along blood vessels, a brown-black discoloration in blood vessels caused by hydrogen sulfide gas, blisters and skin slippage, loss of hair and nails. Putrefaction happens quickly under conditions like excessive heat, illnesses like peritonitis, where there are already too many bacteria present, and places like sewers where there are lots of external germs. Putrefaction can be stopped completely by freezing, or can be slowed down by cooling the body. There are 4 general stages of putrefaction. Putrefaction, which occurs 4-10 days after death, autolysis occurs and gases and discoloration start. Black putrefaction, which occurs 10-20 days after death, exposed skin turns black, bloating collapses and fluids are released from the body. Butyric fermentation, which occurs 20-50 days after death. The remaining flesh is removed. Butyric acid is formed, fermenting the remains, and the body begins to mold if in contact with the ground. Dry decay, which occurs 50-365 days after death. Decay is very slow now due to lack of fluids. Hair and fingernails fall out. That's all of today's videos. If you like this video, then please hit like and share. Also, don't forget to subscribe to this channel. See you in next video. Till then, bye bye.